The Cold Spring Harbor Laboratory was the site of our next great discovery as well. It happened here in 1952. Biologists Alfred Hershey and Martha Chase were studying bacteriophage, a virus known to infect bacteria. Through a process called transformation, the virus takes over the bacterium's internal machinery and commandeers it to produce more viruses. Bacteriophage consists of two simple components, a protein shell, and inside, a mysterious substance scientists had known about for decades called deoxyribonucleic acid, or DNA. Hershey and Chase wanted to know whether it was the protein or the DNA in the virus that carried the genetic information. Using two radioactive chemicals, they labeled the protein in one set of bacteriophages and the DNA in another. Then they exposed a culture of E. coli bacteria to these viruses. After using a kitchen blender to separate the bacteria from the empty viral protein shells, Hershey and Chase saw that the radioactive protein was not penetrating the bacterial wall. But inside the bacterium was the radioactive DNA. It was the DNA, not the protein, that was able to direct the host cell to make new viruses. It was an extraordinary breakthrough. Thanks to Hershey and Chase and the other scientists who helped pave their way, suddenly we understood the true identity of DNA. It's genetic material, the blueprint of life. And it all came out of a blender like this. Hershey was awarded the Nobel Prize in 1969 for his work. But Hershey and Chase's blender experiments went on to inspire a new era of genetic research, beginning with our next great discovery. As late as 1951, the chemical structure of DNA remained a tantalizing enigma. At Cambridge University in England, biologist James Watson and physicist Francis Crick had been working to unlock the secrets of the DNA molecule. But they weren't alone. Several other groups of competing scientists were hard at work trying to solve the same puzzle. Some important facts about DNA were already known. For instance, scientists knew that DNA was composed of 4 bases: adenine, cytosine, guanine, and thymine. They also had been able to infer something about its structure with the help of X-ray crystallography. This technique involved passing an X-ray beam through a crystallized DNA molecule and capturing a vague, shadowy image of its internal structure on a photographic plate. Armed with this information, along with their own knowledge of chemical structure, Watson and Crick began building a 3-dimensional model of DNA.
I wanted an arrangement, where I had a big and a small molecule, and somehow you had to form link bonds. Here's A, and here's T, and I wanted this hydrogen to point directly at this nitrogen. So I had something like this. Ooh. So then I went to the pair. I wanted this nitrogen to point to this one. I went like this. Whoa.
Today, you can buy a kit and assemble the structure that Watson and Crick put together. But we're getting ahead of ourselves. Watson and Crick needed more data. With the help of physicist Maurice Wilkins, they gained access to an X-ray picture of a DNA molecule that had been taken by his research partner. Her name was Rosalind Franklin. Without her knowledge, Watson and Crick used the data from that X-ray image and successfully completed their model. In a 1953 issue of Nature magazine, Watson and Crick revealed their amazing discovery to the world. Their model showed that the DNA molecule was a double helix. The twin strands were composed of pairs of the 4 known bases linked together by hydrogen bonds. and the whole structure corkscrewed like a spiral ladder, which could easily pull apart in order to make copies of itself with the same encoded genetic information. Watson and Crick had won the race. The discovery of the structure of DNA sparked a scientific revolution. It illuminated the molecular and biochemical foundation of life in a whole new way. It opened doors for areas of research and other great discoveries that few ever imagined possible. As for Watson and Crick, their discovery of the double helix, along with Maurice Wilkins, won them a share of the 1962 Nobel Prize. And what of Wilkins' colleague, Rosalind Franklin? Despite her contribution to the discovery, she wasn't considered for the '62 prize. The rules state that it can only be awarded to a living recipient. Rosalind Franklin died in 1958 of ovarian cancer, most likely caused by exposure to X-rays.